Ravinder Garu, and Anuradha Madam, Dean Faculty of Science, and all other participants of this webinar. I am very much delighted to take part in this international webinar on cutting-edge technologies in molecular immunology, organized by the Department of Microbiology, Chaitanya Dept University. As you all know, that universities are meant for generating ideas, supporting ideas, and spreading the ideas. Universities are also supposed to conduct challenge-oriented, use-inspired, practice-led translational research. Universities are also required to undertake extension activities. With regard to our Chaitanya Dept University, Chaitanya Dept University is the first of its kind in Telangana state. Chaitanya Dept University wants to achieve excellence through innovation and quality education. It wants to create responsibility to build a future that posterity can be proud of. It wants to become a center for power of academics and flavor of ethics. The vigor and rigor of Chaitanya academic activities adds power and flavor to future generations. Finally, Chaitanya intends to impart world-class and value-based education with knowledge architecture and technology infrastructure. Now, in other words, universities are supposed to serve the society through their scholarly activities. Now, I believe organizing these seminars is a part of intellectual exercise wherein the knowledge is shared with others. And I believe that teachers receive as much as they give, and as much as they share their views with others, and as much as they put in efforts in learning new things. Our microbiology department is going in this direction, that is sharing of knowledge internationally and adding new knowledge to the existing body of knowledge. I am of the opinion that knowledge could be in the form of a concept, theory, principle, logic, formula, method, technique, model. I hope the seminar would throw new light on the chosen area and also identify the pertinent problems in that area and provide pragmatic solutions in that area which are useful to the organizations, nations, and societies. Recently, Chaitanya University has organized an international virtual conference in collaboration with Research India Foundation on disciplinary, interdisciplinary, and multidisciplinary areas in almost 13 tracks, and it was well received and well appreciated. I wish this seminar also will be very successful in achieving the purpose for which it is organized. I also expect the deliberations during this seminar will be thought-provoking, innovative, and creative efflorescence and useful to the academic world. I suggest the organizers that the deliberations should be brought out in the form of book so that it will be a permanent record for us. We are also proud of our alumni, Dr. Divya, Priya, Ravirala, and Dr. Mukesh Pasupuleti, who are participating as resource persons in this seminar. I congratulate Professor Anuradha Madam, Dean Faculty of Science, Professor Sreelatha Madam, organizing Convener, organizing Secretaries, Manoj, Kumar, Reddy, Jeevan, Chandra, and Veena for their relentless efforts in organizing this seminar. Lastly, I conclude by saying one should possess academic will, research zeal, extension will, and governance skill, and ultimately work for the achievement of excellence and everyday greatness, which are considered to be the ultimates under the sun. I wish all the best and grand success for the seminar. Thank you, one and all.
Thank you very much, sir. Thank you very much. I now request our registrar, Professor Ravinder sir, to give his message on this occasion. Over to you, sir.
Thank you, madam. Good morning. I am very happy to learn that the Department of Microbiology, Chaitanya deemed to be university, is organized.
Hello, ma'am. Hello, ma'am. There seems to be an interruption.
Yeah, ma'am. Just wait a second. Some net issue, madam. Just wait a second.
Okay.
Hello.
Sir. Sir, okay, continue, sir.
Okay, ma'am.
Because of power problems, okay.
Okay. Good morning. I am very happy to learn that the Department of Microbiology, Chaitanya deemed University, is organizing an international seminar on cutting-edge technologies in molecular immunology. The theme of the seminar is relevant since it focuses on the challenges and issues in microbiology to create a research ambience among the students, research scholars, and faculty members. It is a great pleasure that the potential academicians and the persons from industry will be taking part in the conference. I am of the opinion that the deliberations in the conference will enable the participant to solve the unknown challenges to provide us a better future. I congratulate the staff, research scholars, and students of microbiology on the eve of organizing a seminar, which provides an opportunity for the fruitful interaction during the deliberation. This helps us to acquire the knowledge and skills on the theme of the seminar. Finally, I wish the seminar a grand success. Thank you.
Thank you, sir. Thank you very much.
Sorry, sir, there was some issues. Ravinder, sir, you can continue.
Madam, sir, we completed.
Sir, complete chesara, madam?
Completed, madam.
Okay. So I now request Jeevan Chandra to introduce our first speaker.
Thank you,
Jeevan
madam.
Chandra, sir.
Thank you, madam. Am I audible?
Yes, sir. You can continue.
Thank you, madam. I feel it is a great privilege to introduce Dr. Priya Ravirala. Dr. Priya Ravirala is happened to be our alumni, 2004 MSc outgoing batch. She has completed her master's in microbiology in 2004 itself in Chaitanya Degree and PG College, India. Later, she earned her research degree in PhD from University of Houston in Cell and Molecular Biology in May 2021. She has published several journals, international and national journals. She has 12 years of research experience in translational research in the field of oncology, hematology, and solid tumors and immunology. She also happened to be manager several investigation studies in development of cell-based and drug therapies at multinational pharma and biotech companies. She also conducted several scientific publications and extensive lectures in oncology. She also well versed in FDA and European regulations of ICH guidelines. She also completed her core competencies in oncology, immunology related product knowledge. She also gained her knowledge in regulatory affairs, especially in oncology and related products. In the University of Houston, currently she has been the researcher at the National Institute of Health, US. Currently in the University of Houston, she executed studies on improving the efficacy of oncolytic viruses for treatment of solid tumors and designed several investigations on novel chimeric enagers like bicep, tricep for combinational use of virotherapy, and also characterized immune landscape for tumor microenvironment to understand the oncolytic virotherapy. She also completed her research in high throughput genomics, as well as she supervised several projects on to the postgraduate students and undergraduate students at NIH Houston. Not only that, she also extending her scientific services as a senior scientist at a Reneuron as a lab manager in UK in 2013. Later, she also extended her services as a assay development scientist in GlaxoSmithKline Beecham. And later, she also acted as associate scientist in Novartis, Houston, UK. She now also extending her services as assay development for Pfizer and University of Manchester as a research assistant. This speaks her scientific competency, and she also published national, international journals. 4 are getting best publication awards. This is about Priya Ravirala, Dr. Divya Priya Ravirala. Thank you, madam. Over to you, madam.
So thank you, Jeevan sir. So Divya, the floor is now yours. I'm glad to the students who are listening to me. I'm very proud to say that she is one of our alumni member, and we are very proud that she is doing a very yeoman service and also doing the best research that is going on in the world. I now welcome Divya Ravirala to give her message, to present her paper. You can share your screen, Divya. I have already put the screen for sharing. So you can share the screen and the floor is open to you.
Good morning, ma'am. Are you able to see my screen?
Yes, we are able to hear you as well as see your screen.
Wonderful. Yeah, first of all, thank you all for a nice introduction. Thank you, Jeevan sir, and thank you, ma'am, for having me here. It's a great honor to be here for the international seminar on molecular immunology organized by the microbiology department at Chaitanya Degree and PG Colleges, Hanamkunda. And I thank Krishnamacharya sir for valuable words in the beginning of the seminar, and all other professors, Manoj sir, and it's a great honor to share the stage with Dr. Mukesh Pasupuleti sir as well. You've got an introduction to my research and background from Jeevan sir.
As well.
Can you all hear me? Yes. So moving on to my talk, I'm going to present to you about a topic that is of great interest, and that is my research study at University of Houston. So I'm going to talk about synergistic combination of oncolytic virotherapy and immunotherapy for the treatment of solid tumors. So there are basically two parts to my talk today. In the first part, I'm going to talk about the changes in the tumor immune landscape upon treatment with an oncolytic virus. And the second part of my talk is going to be about a strategy on how to engage immune cells within the tumor microenvironment for increasing the efficacy or against the tumor. And this is an immunotherapeutic approach that is combined with combination of oncolytic viruses, and how this combination has given synergistic benefits for the treatment of solid cancers. So if cancer research is of interest to you, and if you're really passionate about cancer research, and this is the talk for you all here. So for any study, as we have a rationale behind each research study, right? And therefore the rationale behind my research work is that combination therapy, which is a treatment modality that combines two or more treatment agents, is a cornerstone for cancer therapy. So the future of immuno-oncology drug development is positioned in combination therapy, where the immunotherapy modalities are tested in rational combinations with other targeted agents for synergistic effect in terms of enhanced efficacy, as well as improved tolerability and long-term survival. So the agent that I have used in my study are oncolytic viruses. So just a brief introduction to what oncolytic viruses are. As we may all know, as you're going probably have studied in the Department of Microbiology here at Chaitanya. So oncolytic viruses are self-replicating, tumor-specific, and they directly lyse the tumor cells. So they're of special interest for us because of their capacity to induce immunogenic cell death. So what exactly happens when a virus infects a cell? So upon infection, viruses hijack the host cell to make viral proteins, of course, for its own survival, without which it cannot survive outside the cell. And when this virus replicates within the cell, it eventually kills the cell, and therefore the cell is lysed. So these dying tumor cells release pathogen-associated molecular patterns called PAMs or DAMs, or tumor-associated antigens, and they encounter antigen-presenting cells such as dendritic cells in the tumor microenvironment. And the viral infection also causes local inflammation, which also stimulates other dendritic cells. And these mature dendritic cells, then they travel back to the lymph nodes and present the tumor-associated antigens to the T cells. So T cells are immune cells which act against the tumor, and this may elicit an anti-tumor CD4 positive and CD8 positive T cell responses. So overall, the T cells can actually now kill virus infected as well as the non-infected tumor cells. So overall, what's happening here is upon viral infection, there is conversion of cold immunosuppressive tumor environment into a hot inflamed tumor, where we see an influx of various different kinds of immune cells, including, as I mentioned, T cells, NK cells, macrophages, dendritic cells. Whereas in a cold tumor, there's lack of these important immune cells. So therefore, the tumor continues to grow. But upon oncolytic viral infection, because of the increased influx of the important immune cells which act against the tumor becomes vulnerable for any other immunotherapeutic modalities that can be applied for the treatment of this particular cancer. So having to know that, we asked ourselves a question, how does Fuson H2, which is an HSV2 oncolytic virus, alter the immune landscape in the tumor microenvironment? So the scientific community overall around the world have turned their focus into the immunogenic properties of the virus rather than its direct anti-tumor effects. So the same effort, I wanted to kind of understand extensively how Fuson H2, which is our lab's HSV2-based oncolytic virus, modulated the tumor microenvironment. And for that, we have used a technology called single-cell RNA sequencing to characterize every single cell that has infiltrated into the tumor upon treatment with the oncolytic virus. So what is Fuson H2? I wanted to kind of start off talking about this virus and how it actually infects the cell. So in normal HSV2 infection, the ICP10 gene of the virus binds to and phosphorylates the GTPase activating RAS GDP and turns on the RAS activating pathway. So the RAS activation is important for the replication of the virus, without which the virus won't be able to replicate in the cell. So we hypothesized that the deletion of a PK domain in the ICP10 gene would eventually render the virus attenuated. So therefore, the virus won't be able to activate the RAS pathway in mitotic cells, non-mitotic cells, and therefore won't be able to replicate. However, as you may know that tumor cells have constitutive RAS activation. Therefore, irrespective of the deletion, the virus would continue to replicate in malignant RAS-activated tumor cells. So as I mentioned earlier, that I've used single-cell transcriptomics to study the tumor microenvironment, and the tumor microenvironment is very heterogeneous. It obviously has a lot of cancer cells that continuously divide uncontrollably. And we have other important immune cells, including T cells, NK cells, macrophages, B cells, stromal cells, and all the extracellular matrix that kind of puts all these cells together in a matrix form. So having to understand that, I want to kind of give you a brief overview to the study design here. So what have we done is we have established a murine colon cancer model, where we have used a CD26 colon cancer line, which has an overexpression for human EGFR. So these cells were implanted into mice, and when the tumors reached approximately about 8 to 10 millimeter size, we have injected the oncolytic virus, which is named as Fuson H2, intratumorally into this subcutaneous tumors in the mice. So after 48 hours of viral infection, the tumors were harvested and they were disaggregated for single-cell RNA sequencing. And at the same time, you also cut the tumors in half to continue for IHC analysis, immunohistochemistry analysis, to validate the results that we observe in the single-cell RNA sequencing. So once the tumor was disaggregated, digested, and kind of made into single-cell suspensions, then they were sorted for CD45 positive cells. So what are CD45 positive cells? CD45 positive cells are immune cells. And it is important for us to enrich for CD45 positive cells when studying the immune cells, because in a cold tumor microenvironment, as I mentioned earlier, there are going to be very few immune cells. So for control, where we have PBS control, we assume that there's a lot more less immune cells. As you can see here in the PBS control, we have very few immune cells, which are CD45 positive here. But whereas in the fusion-treated sample, we have approximately 3% more than what we have noticed in the control tumors. So after sorting the cells for CD45 immune cells, then the cells were combined with CD45 negative cells at the 3:1 ratio, and then we subjected it for single-cell RNA sequencing analysis. So upon single-cell RNA sequencing, we were able to identify various cell types within the population, and we were able to annotate the cells. And this is an unbiased cell type classification where we identified 22 different cell clusters. But the interest was to kind of categorize them into immune cells and non-immune cells. So here, PTPRC, this is another name for CD45. So just it's used interchangeably here. So all the cells here, as you can see, are positive for PTPRC, otherwise called CD45. These are all immune cells. Whereas here we have the tumor cells, which are positive for EGFR. So as I mentioned in my previous slide, the CD26 cell line that we have used to implant the tumors have high expression of EGFR, and that is what we use to identify the tumor cells that we have harvested from this overall tumor treatments. And further classifying the cells based upon these markers, we were able to further subcluster the immune cells into NK cells, B cells, CD8 positive T cells, and CD4 positive T cells, etc. And we have another secluded cluster of tumor cells, which are CD45 negative CD26 tumor cells. So again, here, as you can see, these are all just the CD45 positive immune cells alone. We have excluded the tumor cells out of the cluster here. And kind of trying to understand what changes did the Fuson bring upon injection into the tumor, we kind of stratified the data between the two treatment groups. Here we have the PBS group, which is the control group, and the Fuson group, which is a treatment where the virus was injected intratumorally. So as you can see from the data, the Fuson H3 treatment has resulted in some significant changes in the relative composition of the immune cells. There are a lot more immune cells and various subcategories of the immune cells present in the Fuson treated sample as opposed to the PBS control group. So further looking at the composition between the two treatment groups, as we have identified all these different immune cells, there was a significant increase in the relative composition of the CD8 and CD4 positive cells in the Fuson treated groups as compared to the PBS treated sample, where the overwhelming majority of the CD45 positive cells in the PBS group are monocytes and macrophages. So monocytes and macrophages are innate immune cells, whereas the CD4 and CD8 positive T cells are the adaptive immune cells that play a very important role in the generation of anti-tumor immune responses. And surprisingly, we also found another cell cluster that has significantly increased. So we have indeed noticed an increased influx of B cells in the Fuson treated sample as well. As you can see, there's barely any B cells in the PBS treated control group. And this was very important finding for us because it has become evident in recent years that B cells can be a predictive marker for treatment with immunotherapy options. Right, and as I mentioned earlier, we took half of the tumors for immunohistochemistry analysis, just to kind of validate the results from what we have seen from the single cell RNA sequencing study. So it's always important to rely back on another method to what you saw in a particular experiment. So it's kind of backing up what you actually noticed in your primary assay or primary experiment. So here, when we did the immunohistochemical staining for some important immune cell markers like CD3, CD4, CD8, and CD20. So CD3 is a common marker for all the T cells, and CD3, CD4 positive are the CD4 positive T cells, and CD4 and CD8 are the cytotoxic T cells. So as you can see, the Fuson treated tumors have a lot more staining for CD3, CD4, and CD8 markers, which comprise for the CD4 positive T cells, and also CD20 staining for the B cell subgroup. So these immunohistochemical stainings thus provide a corroborative support for the immune cell clustering that we have noticed from the single cell RNA sequencing studies. So, and next for us, we mentioned, we saw the increase in the T cells, we saw increase in the B cells, but are they in their activated state? Are they ready to fight the cancer? This is an important question that needs to be answered to be able to understand how the T cells perform after reaching the tumor. So for that, I looked at some of the important activation markers. So here we have the CD8 positive T cell cluster and CD4 positive T cell cluster together, and looking at some activation markers here, which are CD28, IL-7R. All of these important activation markers are upregulated in the T cells that have been isolated from the fusion-treated tumors. Not so many with the PBS-treated controls. So this indicates that these T cells that have infiltrated into the tumor upon infection with the virus are indeed in their activated state, and that indicates the responsiveness of the T cells against the tumor. And moreover, we also have some exhaustion markers that we need to look into. And the exhaustion markers are PD-CD1, LAG3, CTLA4, etc. And these markers are downregulated. Activation markers are upregulated, and the exhaustion markers are downregulated, which is a very favorable feature for these T cells or the tumor microenvironment that obviously indicates the responsiveness of the immune system to the tumor. So we are priming the immune system to attack the tumor in a positive way. Not only the oncolytic virus is directly lysing the tumor cells, apart from that, it's also priming or making the tumor vulnerable with all the immune cells that further go on and attack the rest of the uninfected tumor cells that may be still present within the tumor. And moving on, one of the distinct findings, as I've shown earlier, is that the increase in the presence of B cell infiltration in the tumor microenvironment from Fuson H3 treatment. And to further characterize the infiltrated B cells, again, I stratified the data between the two treatment groups. And here there is a distinct difference where we have over 700 plus number of B cells in the Fuson treated sample and hardly about 6 B cells in the PBS treated controls. And looking at the signature markers as I did for the T cells, with the CD79A and 79B are the markers that form part of the B cell receptor, and CD19 and CD20 are some of the typical signature markers for B cells. And we also looked at some of the other markers, functional markers. Again, we wanted to understand how these B cells behaved within the tumor. Are they immune suppressed, or are they in their activated state? For that, we looked at some functional markers like CXCR4, and PAX5, BANK1, which are all important for core receptor activation. And from these analyses, all these marker genes are upregulated, indicating that the B cells are highly functional that have influxed into the tumors that was treated with the oncolytic virus. Okay, so we looked into T cells, we looked into B cells, and for us, the next thing was to look at some other innate immune cells, like natural killer cells, for example. So the natural cell cluster here is further stratified into two treatment groups, and you may not see a difference in the number of NK cells between the two treatment groups here, but in the beginning, if you can recall from the first slide that I've talked about the design of the study, we saw a lot more infiltration of the overall CD45 positive cells. So that gives us that there's obviously more number of NK cells in the Fuson treated sample as well. And again, looking at some of the typical markers like perforin and granzyme. I want you guys to remember this two important molecules, because these are the cytotoxic granules that are released from the NK cells that actually bores holes into the tumor cells and eventually lyses them. And these two important genes are upregulated in NK cells, which indicates that these NK cells are super active. They're cytotoxic in function and they're ready to kill that tumor. And we also looked at some of the other important markers like CD269 and interferon gamma. These are also indicating the activation status of the NK cells. And HCST and KLRK1 are some of the other activation markers that are involved in the cytotoxicity as well as cytokine production in NK cells. So all of these results show that at a higher level, like NK cells are present in Fuson treated sample than compared to the PBS control, are not only more in number, but at the same time, they're also in their activated state. And moving on for the other immune cells here, myeloid cells that comprises of monocytes, macrophages, dendritic cells, etc. And again, I stratified them according to the treatment group. And looking at the composition here, we saw increased presence of neutrophils in the Fuson-treated sample. And this is what we have actually observed in our previous studies as well, where from immunohistochemistry analysis, we've noticed increased staining for neutrophils, and that has been validated in this recent study where we have obviously noticed more neutrophil infiltration into the tumors that have been treated with the virus. And I wanted to kind of further characterize this macrophage or monocyte cluster because there are two forms of these macrophages. One is M1 phenotype, the other one is M2 phenotype. And M1 phenotype is the most favorable type of the macrophages, which are actually involved in the immune stimulation, inflammation, and actually generation of cancer-specific immune cells. So looking at some of the M1 markers, we've noticed that the M1 markers are equally expressed in the Fuson-treated as well as the PBS-treated samples. However, we have seen downregulation of the M2 phenotypic markers from the cells that were treated with the virus, which indicates that the changes that happen upon infection with the oncolytic virus have actually tilted these macrophages towards an M1 phenotype, which is favorable, as I mentioned, for the generation of cancer-specific immune cells. So what you have noticed so far that Fuson H3 treatment induces increased infiltration and actually changes the immune landscape of the tumor microenvironment in a way favorable for anti-tumor activity. So for all of these cells to function in harmony, it is expected that these cells need to communicate intercellularly, like they have to be able to communicate. Innate immune cells have to communicate with the adaptive immune cells and the rest of the stroma or tumor cells to function collectively towards the eradication of the tumor. For that, we did an analysis called cell chat analysis, where we looked at the interactions between these different cell types within those tumors. And as you can see here, we actually noticed more than double the number of interactions in the Fuson-treated sample. And not only that, the interaction strength among these different cell types is also higher compared to the PBS control. And here, as this is just an obvious plot that you can see, Fuson-treated sample have actually all these different cell clusters here, tumor cells, B cells, CD4 T cells, CD8 T cells, and all of these communicated within themselves, but also with the other cell clusters, and extensive communication that is happening within the different cell types here. But whereas there's very fewer interactions in the PBS treated control, and as you can see, B cells are actually secluded out of this communication network. So overall, this indicates that Fuson treatment group generally displayed a higher level of connectivity and increased the number of interactions between various cell types. So here, just to conclude, this is my first part of the talk, from which we can conclude that Fuson H3 treatment can actually convert a cold immunosuppressive tumor microenvironment into a hot inflamed tumor, where you can apply additional treatment applications. So towards that, understanding this modulation of the tumor microenvironment, will also pave new ways for developing potential therapies as well. And to address that, we asked ourselves the next question here: how can we harness the immune modulating potential of these oncolytic viruses to enhance its anti-tumor efficacy? Can we combine it with other treatment modalities? And to answer to that, the answer to that question basically is that yes, oncolytic viruses are combined with other treatment regimens around the world, including radiotherapy, chemotherapy, HDAC inhibitors, etc. But my interest has been combining oncolytic viruses with immunotherapy, where we can actually see increased in pro-inflammatory responses in the tumor cells, increased in anti-tumor activity, eventually leading to the cell death. So for that, I've come with a novel strategy of actually engaging these immune cells with the tumor cells. And for that, I've designed chimeric engager proteins, which I name them as bicep and tricep. So it stands for bispecific chimeric engager protein and trispecific chimeric engager proteins. So in the bicep, where we have two components, therefore the name bi, where we have the OMCP component, which is linked to an EGF molecule by means of a linker in between. And I want you to pay attention to the MIC tag here, which is what is going to be basis for the detection of these proteins in our following experiments. And there's a strong reasoning to why these components have been chosen, which is I'm going to mention to you in the next slide, where I'm going to talk about the mechanism of action. But however, just wanted to kind of briefly give you the design of the molecule. And we did some 3D modeling studies as well after fusing these two components to look for any conformational changes or how the components are spatially distributed in the 3D space. And for that, when we did the 3D modeling studies, there was no conformational constants that happened despite fusing these two molecules together. And in the tricep molecule, we have placed an IL-2 component upstream of OMCP with another linker here. So again, the same thing, we actually did 3D modeling studies, and there's no conformational constants again. And we were able to kind of synthesize these molecules in an in vitro mammalian system as well, as you can see from the Western blot analysis. And the interesting part here, the simultaneous engagement of the immune cells with the tumor cells and how the mechanism of action. So here, as you can see, this is an NK cell or a T cell, a CD8 positive T cells. And these immune cells, NK and T cells, express NKG2D receptor. This is a receptor, which is an activating receptor present on NK cells. And the OMCP component, which is present in the bicep molecule, would bind to this receptor. And this engagement on one side of the molecule, and on the other side, we have the EGF alpha ligand, which would assume that would bind to the EGFR receptor on a tumor cell. So most of the tumor cells have upregulated EGFR expression. So therefore, this simultaneous binding of OMCP to NKG2D and EGF alpha to EGFR will efficiently engage the NKG2D bearing NK and T cells with the tumor cells, bringing the two cells in close proximity, creating an immunological synapse. And this will further activate the NK cells to release perforin and granzyme molecules, which eventually lyse the tumor cell. And this is basically the mechanism of action behind this engagement. On the tricep molecule, as I mentioned, we have the IL-2 component, which would bind to the IL-2R receptor. And this binding of IL-2 to the IL-2 receptor would further activate downstream pathways like JAK-STAT pathway, which would further contribute towards proliferation, survival, and differentiation of this NK cells, which will further help to attack more tumor cells. So this is a proposed mechanism of action for both of the bicep and tricep molecule. And the next thing for us to do is to actually test those molecules. Are they really functional? Are they really binding to their receptor? So to prove our hypothesis, I conducted in vitro assays. These are binding affinity assays, where you actually use two cell models. In the first model, I have used tall 104 cells, which are a kind of NK and T cell hybrid type. They have overexpression of NKG2D, and you incubate these cells with the bicep and the tricep supernatants, and you're looking for the double positive cells here, which are positive for NKG2D as well as the MIC tag that is present on both of the bicep and tricep molecules. So what we are seeing here is that the cells upon incubation with the protein supernatants have got the bound protein on their surface by means of the receptor that they express. And this is on one side of the molecule. And on the other side, a similar experiment was performed where we were looking at the binding of EGF alpha to EGFR receptor on a tumor cell line, which is SKOV3, which is an ovarian cancer cell line. So these cells were incubated with the bicep and tricep supernatants again, and we looked at the double positives. As you can see, the EGFR positive SKOV3 cells have bound protein molecules, both bicep and tricep, as you can see with the double positives. But whereas there are some EGFR negative SKOV3 which acted as a control here, and because there is lack of receptor on these cells, the protein would not be able to bind to these cells. Okay, so the next thing for us to look at is the in vitro efficacy of the molecules, whether or not these cells cause increased cell killing. And for that, we conducted an in vitro tumor killing assay where we incubated the immune cells with the tumor cells at varying effector to target ratios in the presence or absence of these protein cocktails, either bicep or tricep. And we looked at the growth of the tumor cells over time. Here we are looking at a 24-hour time period. And from these plots, we can notice clearly that there was inhibition of the tumor cells upon incubation with the protein supernatants at the different ET ratios compared to the control, which is the mock transfected supernatants. So this showed us the efficacy or increased killing associated with the engagement of these two molecules when in the presence of these two molecules, when the tumor cells and immune cells were co-cultured in a in vitro system. And a similar assay was performed. I'm not going to go into deep details here, but again, as you can see from at effector to target ratio of 1:1, both bicep and tricep molecule have shown efficacy with increased activation of caspase-3, which is the marker for cell death. So we are looking at effector ratios of 1:1, 3:1, and 5:1, whereas at the higher effector to target ratios. So effector cells are the immune cells and target cells are the tumor cells. And we are co-culturing these two cells here. At higher ET ratios, we have seen that tricep molecule is much more efficacious than bicep or the control, which is the non-transfected supernatants. Okay, so we've designed the molecules. We've tested them in vitro for their binding affinities. We also tested for the in vitro killing capacity. The next good thing for us to do is to test them in an in vivo model, in a mouse model. For that, I've used a colon cancer model again, where we use the CD26 cell line that I described earlier and treated with the virus and looked at the tumor growth over time for over 2 weeks here. And as you can clearly see from the plots, I have 4 treatment groups. PBS is the control. Synco2D is the name of the virus, another virus which has the GFP expression, which is an empty virus without the transgene coding for the bicep or tricep molecule. And we have the other 2 treatment groups where we have the virus encoding for the bicep molecule and the virus encoding for the tricep molecule. When all of the mice were treated with various treatment groups, as you can see, the Synco2D, bicep or triceps, significantly prolonged survival, and there's reduction in the tumor growth compared to the PBS control group. So we saw some efficacy with the Synco2D GFP alone, but there is an increased tumor reduction in the mice that were treated with the virus that encoded for these 2 chimeric engager proteins. And these are some of the tumors that were harvested out of the mice at the end of the treatment period. And there's a reduction in the tumor growth that is very obvious from these images. Sorry about that, but I have to show you to prove that there was tumor reduction. So the next thing was actually I wanted to kind of look at the presence of the NK and T cells in the tumor microenvironment because those are the cells that are required for the bicep or tricep to function efficiently. And for that, I looked at the single cell RNA sequencing again, isolated the cells from each of these treatment groups, looked at the composition of the cells. And as here, there are a lot more NK cells as well as T cells in the tumors that were treated with the tricep molecule compared to our PBS and the GFP treatment groups. And later on, the next question that I would ask myself is how well are these T cells and NK cells functional? For that, the important molecules that are very important for killing the tumor cells, KLRK1, granzyme and porphyrin, all of these molecules are upregulated in the tumors that were treated with the virus that encoded for the tricep molecule. So there you can see all of that. Very nicely upregulated expression of all of these important gene markers in the cells that were treated, in the tumors that were treated with the virus, which encoded for these chimeric engager molecules, which kind of supports that not only the oncolytic virus is directly infecting and killing the tumor cell, these chimeric engager proteins would further engage those infiltrated NK and T cells and engage them with the non-infected tumor cells and kind of enable further killing of the rest of the uninfected tumor cells. And again, looking at some of the important immune cell markers as well, we have seen increased upregulation of some of these important marker genes in both the NK and T cell clusters. So summary from the talk is that the first part of have demonstrated that there is an increase in the infiltration of important immune cells, NK cells, T cells, B cells, which basically convert the cold tumors into hot ones. And to kind of engage those infiltrated immune cells of designed chimeric engager proteins. And these chimeric engager proteins, when they're co-administered using the virus, can represent a potential way for increased anti-tumor efficacy and further potentiating these immune cells to attack the rest of the tumor cells. So this is a take-home message where co-administration of these uniquely designed chimeric engagers may represent a viable way of potentiating virotherapy for solid tumors. So combination therapy is supreme over single or monotherapy options. So all the two parts of the study has been published. This work has been published in Journal for Immunotherapy of Cancer. So please have a look at it if you need to know more details. And if you have any more questions, I'm ready to answer at the end of this session, but you can also reach me otherwise, and I can share my email with you later on. I would like to thank some of our collaborators who have been involved in this work. And I thank you all for your attention, and I'm happy to take any more questions or any questions that you may have. And I highly thank Anuradha ma'am for having me here, for inviting me to present my research work, and also all the professors during my master's from 2002 and 2004, Manoj sir, Mukesh sir, and more than ever, Purushottam Reddy sir, who have been very grateful in giving the kind of education that we all needed to grow up in our careers. Thank you so much, ma'am, for having me.
Thank you, Divya. It was a real, and I can say it was a very novel research which you have taken. I just wanted to ask one question. Has it been used in any treatment on human beings?
So we are trying to move our virus into the clinic. It is currently in the clinical trials.
Okay.
We are trying to test the virus in the phase 1 clinical trials at the moment.
Okay. So what further you want to do more research in this particular field?
So cancer research has been my passion, and obviously my focus has been kind of to provoke our own immune system. So our immune system is capable of many things. I mean, you have to kind of just prime it the right way, and it can attack the cancer and clear it. And that is where my interest lies in. And I'm actually exploring further into immunotherapeutic modalities for treatment of basic solid tumors, especially.
So that was very nice, Divya. I request Jeevan sir to just conclude with the rapporteur report. Suman, just stop your share.
Sure.
Madam, am I audible?
Yeah, you are audible.
Thank you, Dr. Divya Priya Ravirala. It's a very elaborative lecture on testing chimeric activity of programmed cells as well as a tumor microenvironment. This lecture will gives a clear explanation regarding tumor microenvironment and where is MSC and biomarkers have been interacting in a
Just continue, Jeevan.
Madam, am I audible?
Yes, you are audible.
Thank you, Divya Priya Ravirala, for giving this lecture. This lecture will tell us the essentiality of a tumor microenvironment where natural killer cells and programmed T cells are interacting with so many biomarkers as like cluster differentiation factors CD4 cells and CD8 cells, how they are behaving within natural killer cells. And this technology also give us the clearance regarding how macrophages will be activated in the signal cascade pathway when M1, M2 population have been behavior with the respective macrophages. In this lecture, it is clear that tissue necrotic factor, interleukin 1, interleukin 2 are both very essential for recruiting the cytotoxic T cells. Furthermore, the fibroblast cells and progenitors, how they are is very important. And moreover, the inhibition of tumor-associated angiogenesis also pave a way to how to arrest the tumor cells in specific manner. At the same time, bicep and tricep chimeric operants, how they have been behaving and recruiting the natural killer cells, JAK start pathway, and recruiting cytotoxic potential signal cascade to targeting the tumor cells has given me a wonderful what you call pathway to develop several chimeric biceps and triceps. Finally, I would like to congratulate Divya Priya Ravirala for giving this essentials of biomarker technology and toll-like receptor 104 importance and recruiting of tumor helper cell 1 and 17 for cell therapy and upregulation of genes like KIRK1 and GZMB and protein F1 also very important. So thank you, madam, for giving this lecture and really your lecture is enlightened us and to discover novel innovative ideas regarding development of tumor killing viruses. Thank you very much, madam, for giving this opportunity.
Thank you, Divya. Thank you very
sir.
much. So
Thank you,
you can
ma'am.
even interact. The next speaker is your own professor, Dr. Mukesh Pasupuleti. So welcome Mukesh sir to this conference. I request Manoj sir to introduce Mukesh sir.
Honorable Vice Chancellor, respected dignitaries, and I'm very fortunate here to introduce Dr. Mukesh Pasupuleti, who happens to be a principal scientist and associate professor CDRA Lucknow. And coming to his introduction, now if I go on reading his efficiencies and achievements, now it becomes a 20-page. I have made it very short. He has completed his MSc in biotechnology from University of Calicut, and then he has pursued his PhD in clinical medicine in 2009 with specialization of immunology from Lund University, Sweden. Coming to a person's efficiency depends upon the publications made, and he has made around 90 publications with total impact factor of 270.16. And citation numbers 3,598 with H index as on August 2021, it is 28, and I-10 index is 60 as on August 2021. And coming to the books published, he has published 2 books, 1 chapter, and both international conferences attended by him are in 30s. He has evaluated 10 PhDs and he has completed one. He has completed as a principal investigator. He has completed 4 projects and one is outgoing. As a co-investigator, he has completed 2 with the 2 ongoing projects. He is a grant reviewer at both international and national levels. At international level, he is a reviewer for Natural Environment Research Council UK, Chilean National Antarctic Program, and also Vivo funding Sweden. Coming to national grant reviewer, is reviewer for Department of Science and Technology, DST India, Council of Industrial Research India, Indian Council of Medical Research. And he is a reviewer for various journals, and these journals include is an expert opinion on drug discovery, infectious disease and therapy, Annals of Microbiology, Journal of Immunology, International Journal of Peptide Research and Therapeutics, Microbial Research, Development and Comparative Immunology, Gene, International Journal of Peptides, Indian Journal of Environmental Biology, Electronic Journal of Biotechnology, Journal of Dermatological Sciences. And he has research experience. He has 5 PhD programs completed under him, and 3 are pursuing, and MSc projects, 11 MSc projects, 3 international and 8 national. Postdoctorals, 4, and he is a PhD guideship eligibility in JNU New Delhi, ACSIR New Delhi, SRM University Chennai, Manipal University Manipal. He is a PhD evaluator for Osmania University, Kakatiya University, Lucknow University, and SRM University. SRM University and also IIT Roorkee. And coming to the prizes or awards, certificate fellowship, he was selected as senior scientist about 2016 by Association of Biotechnology and Pharmacy, Guntur, Andhra Pradesh, India. Received Outstanding Scientist 2016 award from Venus International Foundation, Chennai, India. Received Dr. ABJ Abdul Kalam Award for Scientific Excellence 2016 award from Marina Labs, Chennai. Selected as the outstanding student at faculty performers in their pursuit of biotechnology opportunities and options 2015 by the Department of Biotechnology, Government of India. Selected by the Department of Biotechnology, Government of India as the High Achievers Trailblazer in the Biotech Career Reddy Roorkee, Recon 2014 by the Department of Biotechnology, Government of India. He has received travel grants for the Faculty of Medicine, and he has received scholarships for 2 years, July 2018 to 2000, from University of Calicut during his masters. And affiliations to scientific organizations, he's a member in both international and Indian organization, life member of British Society of Antimicrobial Chemotherapy UK, life member of American Peptide Society USA, life member of International Society for Infectious Diseases USA, life member of European Peptide Society, member of American Society for Microbiology, life member of Society of Biotechnologists, life member of Indian Peptide Society, life member of Indian Immunology Society, and life member of Indian Society of Cell Biology, National Advisory Committee member of Indian Nanobiologists Association, life member of Society of United Life Sciences, Souls, and managing advisor for Biotech Dhaba. And Board of Studies member, he happens to be Board of Studies member for Institute of Engineering and Technology, IET Lucknow, and Chaitanya Institute of Technology and Science, that is Chaitanya deemed to be University, Warangal. And moreover, the most important thing is he happens to be one of my colleagues, and I am proud to say that. And moreover, the important thing is he again happens to be the alumni of this university. And welcome, Mukesh. Thanks for your grand presence at this international seminar. Thank you.
Thank you, Mukesh, sir. He's also the Board of Studies member for the Department of Microbiology also. So Mukesh, sir, you can share your screen and you can start your presentation.
Okay. Thank you for having me. It's nice to meet all my students and my faculty colleagues and my friends. So I think last 2 years of pandemic has locked us, all of us, but it's nice to meet you all people. So Thank you, Anuradha ma'am, for inviting me for this lecture. So thank you very much. And thank you, Manoj, for a nice introduction. So today I would like to talk on our own work that we have been pursuing from last 15 years, like how we can use immunomodulators or immune components as drug molecules. I would like to start with my own work. Some issues there. So I would like to say a brief about our core research. We work on 4 different angles like host defense peptides, immunomodulators, and we work on quorum quenching and quorum sensing mechanism in the pathogens, and we try to make antimicrobial agents for using small molecules. Our group entirely focus on developing the antimicrobial agents, and we focus not on all the pathogens, but we focus on 5 group of pathogens, that is the escape group of pathogens. So before I start, I would like to tell you what is an AMR. AMR is nothing but a resistance mechanism against an antimicrobial agent developed by a pathogen. So cancer cells can also have resistance, and we call them as resistance. But when we say antimicrobial resistance, then it becomes very specific for the bacteria, virus, fungi, and parasites. So why we are interested in it is for a couple of reasons. One of the reason is it is predicted that by 2050, there will be an average population mortality will be 10,000 population per day. And that is going to be a significant factor in India. In India, it is especially in Asia, their prediction is that there will be a huge number of deaths. And why we are focusing on antimicrobial is we have stopped the antimicrobial resistance discovery in way back in 1980s only, and whatever antibiotics that we now use are the modifications of the previous one. So what are the 4-5 pathogens that we focus on? We focus on Enterococcus, Staphylococcus, Klebsiella, Acinetobacter, Pseudomonas, and Enterobacterial species. So these are the 5 pathogens that we work on, and the reason why we work on this is these pathogens are on the top priority list of WHO. The WHO says that these are the 5 pathogens which account for 80% of deaths that happen around the world when it is coming to the infectious diseases. So what is the disease burden? Because I work in a government organization where our primary mandate is to work for the poor, where we work for drug development programs for the people who cannot afford to heavy costly drugs that are being made in foreign countries. So before we start our work, we started to look at what is the best area to work on and what is the disease burden. And when we looked at it, that we found that the 16% of the world population live in India. Out of the total mortality, 18% of mortality is contributed by the Indian population, and 20% of the morbidity is contributed by the Indian population. But when we look at the amount of the money we spend on infectious disease treatment is very less. And when we look at the healthcare investment, it is also very less. So we thought, okay, we'll work on this area as antimicrobial resistance. So next we took out of many diseases, should we focus on infectious diseases or not? Because after you work for 20 years and you realize that the disease does not exist, then your 20 years of work is gone. So we looked at it. How is it going to be in the next coming 10 years when I started my group in 2014? So we looked at the profile and we realized that the infectious diseases are going to rise very soon, and they contribute around 20% of the deaths that happen all around the world. And if you look at the recent releases by the WHO, there are many programs that have been working out on infectious diseases like antimicrobial resistance program and like what should be done and what should be the surveillance on antibiotic consumption and what is the global action plan. And now CSIR has started a big program called AMR. It's still going to be we are waiting for the funds to come, and once we have the funds, we then will start a huge level of antimicrobial resistance screening programs. So when we started, once we decided that we will be working on antimicrobial resistance, so we went back to the drawing board and we started to look what should be the molecule, because small antibiotics have been discovered primarily in 1940s by the bacteria. But at that time, resistance was not taken into consideration, nor people have asked a question, okay, this bacteria is making this antibiotic, but how is it protecting itself? If that question was asked, we would have been in a different position today. At the time when the discovery of antibiotics, no one made this question like, okay, this bacteria is, or this particular actinomyces is making the antibiotic, how is it protecting itself? So we don't want to do that mistake again. So we went back to our drawing boards and looked at different molecules, how people are working on what are the angles that people are working on. So we looked at different angles, and some people are working on the redesigning of antimicrobials. Some people are working on using old drugs in new formulations or something, and some people are behind natural products for antimicrobial agents, and some people are using target-based approach, and some people are making in the labs like synthetic approach. So we thought like, okay, what should be, okay, these people are working, but how many people are working on, in which area of these people are working on? So when we looked at the profiling of the people or the various literature, we found that out of the 407 clinical programs that are at the clinical stages, 46% of them are working on small molecules, and the rest 54% are working on different other molecules. So then we looked at different programs, and we found out that immunomodulation, people have not explored very much of using immunomodulators as novel antimicrobial agent. So we thought to explore them because you have to, when you start your own group, you don't find with bigger groups, so you have to find your own niche and your own area. So we thought like, okay, we will work on immunomodulators where we want. So when we decided that we will be the other reason why we wanted to work on immunomodulators is for two reasons. The first thing is, as I told you, that antibiotics have antibiotic resistance was not looked critically during the early stages of antibiotic revolution. But now we know that, okay, if a bacteria is exposed to a particular type of molecule, it has a resistance mechanism. Like a lot of people are talking about, they will use Casper technology, they will use bacteriophages to kill bacteria. And some people are trying to use different molecules, but they have to realize that bacteria are also smart organisms. They have their own resistance development mechanism. Like if you use bacteriophages by using CRISPR-Cas9 or CRISPR technology, they can protect themselves. If you are using nanoparticles, then they can use ion efflux pumps, or by membrane metal resistant operons, they can resist the nanoparticles. Like if you use small molecules, then they can use biofilms and extracellular polymatrix, or they try to modify the target site by single mutation. So we thought like, okay, these are the technologies that will fail if not today, in coming line. So we wanted to look on different angle. So when we looked at the other thing that catched our attraction was like the peptides and proteins, which have been slowly being used up. So we focused on what all people are doing in the area of peptides and proteins. So there are a lot of people are working on monoclonal antibodies, Sedero4 conjugates, and efflux pump inhibitors, and some people are using antibiotic enhancers, that is to block the efflux pump so that the antibiotic stays inside the body for longer time. and a lot of people were working on. So when we looked at this, we decided to go for the immune protection, because when we looked at the evolution, we realized that evolution and bacteria co-evolved together. Even though the day human species was born on this planet, the bacteria started colonizing the human body. Like if you look at your human body, about 80% of your body is occupied by the bacteria, and whereas the other 20% is what is human cells. So if I count all the cells, there are around 30 trillion cells in my body, but there are 100 trillion bacteria on my body. So we thought like, okay, bacteria is present and the body is present. So how are they coexisting, or how they are fighting? Then we segregated the bacteria that is present on the body into two components, like the useful bacteria and harmful bacteria. So the useful bacteria is what we call it as today in the term as microbiome, where we always look at what is the good bacteria that we need for the happy survival or a healthy survival, and what is the bacteria that causes a disease. So we asked the first fundamental question, okay, the bacteria is present on the body, so how are we protecting ourselves every time? So we looked at the two components of immune system. The immune system, for a dialectical reasons, your teacher might have taught you, or most of you have studied in this way, as innate immunity and adaptive immunity. Technically, there is no boundary line to say that this is innate immunity and this is adaptive immunity. It is just for dialectical and for easy understanding, it has been segregated, but there is nothing. But function wise, innate immunity components like complement system, NK cells, dendritic cells, these work very fast. They come like once the bacteria invades the human body into the inner tissue, the neutrophils start functioning, and then the phagocytes come, and then you have your own reactions going on. And epithelial barrier is one which protects your body 24/7 from the pathogens. So we thought like, okay, innate immunity is one of the line. So we thought like, when we have looked at it, innate immunity or adaptive immunity, which one to work on? So we thought like, okay, we'll work on innate immunity because this is a fast acting one, and like antibiotic also act very fast. When you have an infection, you need to clear the infection as soon as possible. You cannot say that, okay, if I take a medicine, it will start working after 2 days. No, it has to work very fast. So we thought like, okay, we focus on innate immunity components. So then we dissected like, what are the components of innate immunity system and what should we work on? So we looked at the innate immune system and decided that we will be working on host defense peptides, or the small molecules in the entire system, because you have the mast cells, neutrophils, natural killer, and these all the components of innate immunity. But host defense peptides were less explored at the time. So why we wanted to focus on innate immunity or host defense peptide were for two fundamental reasons. Like when you take a vaccine protects you against a single pathogen. Whereas if you give the patient a host defense peptide, it protects the individual from multiple pathogens, because it is a non-specific, non-memory, and a fast-acting system. So we thought, okay, we'll because these are the molecules which act very fast. So why don't we develop these molecules as biotherapeutics or biological molecules, biological antimicrobial agents? So we call the term biopharmaceutical because they are basically biological in nature, unlike small molecules. And there are different people have used biopharmaceuticals for different terms, like some are called DNA-based drugs or RNA-based drugs or wholesale products, and these all comes under biopharmaceuticals. But these biopharmaceuticals are basically proteins or DNA-based molecules. They're basically biological in nature. So why people are very much interested? If you look at the recent FDA approval, 1 out of every 4 new drug that is coming into market is a biopharmaceutical. That means 25% of the drugs that are currently in the market are basically biopharmaceutical based. So why people are so interested is because they are very, very highly effective, highly specific. They have very few side effects, and they are not, they do not induce carcinogen. cancer in the body, and they're very easy for commercial production by using gene technologies or by genetic engineering. But one of the major drawback of the biopharmaceuticals is they're a bit expensive. They are still not in the reach of poor. So we are also looking into that, how to reduce the cost of it. What are these biologicals or biomolecules? These are normally molecules which are in the size of 10 to 100 nanometers. And these are basically proteins or peptides in nature. And they're used for treating various health disorders like infections, cancers, viral infections, arthritis, and everything. Like different people are now showing very much interested in biopharmaceuticals because of their unique high efficiency and high specificity. So what is the fundamental difference between the biopharmaceuticals and small molecules that are commercially available? It's in the size. Small molecules are very small, like they have a molecular weight less than 500 Daltons, and whereas biologicals have molecular weight starting from 1 kilodalton to 10 kilodaltons, and their structure is a bit complex, and they are produced by the living cells. They are not chemically produced in the lab. And they're one of the major drawback why they are not in the reach of poor is because of their storage and handling issues. They need to be stored at 4 degrees or sometimes at -20. And this is one of the cost factor. But if you look at the patenting thing, out of recently, the patents have been very much filed up in the area of biologics, like almost all, like 2 times the small molecules. If 100 small molecules are patented, 200 small biologics have been patented, and this is rising, and that is why. So biological therapeutics are grouped into 4 classes, like group 1, group 2, group 3, and group 4. And we work on group 1, which is a peptide therapeutics with regulatory activity. We don't work on viruses, we don't work on antibodies, we don't work on diagnostics, because you cannot work on multiple things. You have to have a focus on what you are doing your work. So coming back to our major work that we do on host defense peptides, are basically they're small molecules of 12 to 50 amino acids, and they have a positive charge of 2 to 7. And one of the important thing is that these AMPs, or host defense peptides, are present in almost all living organisms. From microorganisms to humans, everyone produces the host defense peptide. Mostly you might have heard the word called antibiotic, which is produced by the bacteria, whereas in the humans we call them as host defense peptides. And there are 4 different classes of host defense peptides, like helical, beta sheet, and random, and mixed culture. So these host defense peptide work on bacterial membrane. They are not very specific for any target. They don't work like inside the bacteria, but they work on the bacterial surface. So simply they bind to the bacteria and they create pores. Because of the pores, the entire cytoplasmic content comes out. And this is the mechanism how they work. They are not very target specific. And one of the other reason why host defense peptides have been gaining a lot of attention is that they have multiple functions. Like they help in wound healing, they help in the migration of neutrophils, they help in the reducing the inflammation conditions, and they sometimes even help it in the recruitment of cells, so that the second wave of infection does not happen. So this is one of the reasons why people are working on host defense peptides, and why the patents have been increasing in this number. Because if you have a molecule, you can look at different functions and try to make a therapeutic out of it. So your success rate is very high. So host defense peptides, normally characteristics are very simple. They have a hydrophobic amino acids more, and they have mostly charged amino acids like lysine, arginine, and histidine. And they have a different structure potential. That means they can have random or coil or helix. And one of the important point is when they bind to a lipid surface, they normally assume a helical structure, which creates the pores in the body, in the bacterial surface. Other important thing is, as I told you, there are 4 classes like helical peptides, beta sheet, and alpha helices, and mixed culture. Out of them, we focus on 2, like the looped structures and helical structures, because these are easy to synthesize. And when it comes to drug development, or when it comes to the biopharmaceuticals or anything like drug, it is the lesser are the steps in your synthesis, the lesser are the complexities in your synthesis, the better is the molecule to make it as a commercial. Because the more, if you have more complexes, then you have a lot of troubles at the downstream. That means when you synthesize them on a large scale, or when you synthesize them on a transportation, you have some issues which have to be sorted out at that stage. So better you plan your experiments or plan your molecules as early as possible. So that's why we focus on 2 things, which are like the alpha helices and the random one, or we call them as looped structures, so that we have lesser issues at the later stages. Just give me a second. Hello. पांडे जी, मैं एक मीटिंग में हूं, आपको दोपहर में पेपर्स दे दूंगा. Okay. Sorry for the trouble. As I was telling you, we focus on these 2. So the first question is like, how do we do the biopharmaceuticals, or how do we develop? So the first part is we try to screen the genomes in the natural databases, that like the whole genome database. We try to screen for various proteins or peptide, like 50 amino acid stretches, and try to see where are the epitopes, like the high hydrophobic domains or the high hydrophobic charges, where are the high hydrophobic amino acid stretches and everything. And then we try to make peptide those 50 amino acids, and then we try to screen them in vivo or in silico by using different programs. Like how much is the hydrophobicity, whether the hydrophobicity is at the 3rd, 6th, and 9th position to form a perfect helix, or whether it is at the N terminal and C terminal is very free, like where are the charges. So we have some set of criteria, like 50 criteria points, and based upon that we try to screen the peptides and sort out the false positive and potential hits. And once we identify them, we try to look at their structure, how they are forming the structures, and based upon the structures, we further shortlist them if they form into helix, like the alpha helix or the random coil, which is our area of work basically. So once these peptides are there, then we synthesize them by using peptide synthesis, and then we try to do the in vitro assays to determine the activity. Once the activity is decided, whatever molecule works against the bacteria, we try to do a QSAR modeling, and we try to get the better understanding how the peptide will work. So what is the, how do we decide which peptide will go for synthesis or not, is on these parameters, like what is the hydrophobicity, whether the hydrophobicity is 50 to 60% or not, whether the Agadir is there. Agadir is the nothing but the propensity to form the helix. And what is the tilt angle? Tilt angle means how much the peptide can tilt like this. So whether it can tilt or not. If it does not tilt, then sometimes what happens is the peptide does not enter deep into the bacterial surface to cause a pore. And then we look at the net charge, isoelectric focus point, and different things. We try to look at them. And we always try to make amino acids which are less than 18 amino acids stretches, because the most thing is we always keep in mind the cost effectiveness. The smaller is the peptide, the better, the lesser is the cost. The bigger is the peptide, the more is the cost. So once we all, based upon all these parameters, we try to shut down from like 3000 peptide sequences, we come to like 20 or 30. So it's a huge work, but it's worth spending time on it. So once we identify what are our lead molecules or what are our peptides that we want to work on, then we synthesize them, and then this is how we do it. Like we do the preliminary antimicrobial screenings like MICs, and then we try to do these cytotoxicity profile, like how whether they're toxic to human cells or not, and then we try to see how the peptide works on the clinical isolates, how it works on the serum, and then we do the ADME studies. ADME studies means how long the peptide is stable in the plasma, how is it binding to the plasma proteins, how much, whether it is killing the RBCs or not, and these all tests we do. And then once we do all these tests, then we have a go no go decision tree, and based upon once the molecule is say that it is safe and to go on, then we do these studies in the mouse, like PKPD and efficacy studies in mice, whether it is working in the mice or not. And then we try to see whether the resistance development is occurring or not. This is the recent step that we added now. And then we try to do what is the effect of the host defense peptide on the human body organs. Like we try to use different cell lines to see whether anything is happening to human organ. And once we are done with that, then we go for the full-fledged animal studies in different 2 species, like in the rat or mice, and then we try to do the full clinical development. So the first step that we try to do is high throughput screening. Like here is an example where we try to see how is the activity. Like we have some peptides which have given positiveness in our initial in silico screening, but when we put the peptide on the bacteria, it did not work out, but the other peptide worked on. Like we always, as I told you, that we focus on escape pathogens. So we have taken Staph aureus and Pseudomonas, 1 gram positive and 1 gram negative. And then we did the activities like how did they work in the salt conditions and in plasma. And then whether they have any cytotoxicity or not for against the RBCs. So our data shows that one of the peptide like LQF15 works better against clinical pathogens. So, and then we did the various analogs or various modifications, and then we did a MIC. MIC is basically a method where we identify at what is the minimum concentration at which 99% of the bacteria are killed. So normally this, the lower is the value, the lower is better when it comes to MIC. And once we do the MIC, we always do the cytotoxicity and try to find out at what concentration the peptide is showing the activity. And based upon these two, we try to calculate the therapeutic index. This is what is the most important thing. Therapeutic index is nothing but it is the ratio between the toxicity and the activity. And this is the value, the higher is the therapeutic index, the better is the molecule. How much bigger the therapeutic index is, the better is a molecule. And this is what the government regulatory organizations ask, like, what is your therapeutic index? They don't ask you what is your MIC, what is your cytotoxicity. They ask you what is your therapeutic index. Therapeutic index is nothing but the ratio between the toxicity versus the activity. Or in normal terms, we call them the therapeutic index. We normally call them as the risk versus the benefit effects, benefit versus the risk effect. And then we always try to do the cytotoxicity. So how do we do cytotoxicity? We always do MTT and LDH assays. LDH is lactase dehydrogenase enzyme, which usually stays in mitochondria. But when the cells are damaged, this enzyme comes out. That means the peptide is, as I told you, that peptide acts by the membrane damage. So they normally kill the bacteria. So based upon this, we observed that our peptides are not toxic to the human body, but they still have a better antibacterial activity. So then we investigated how it works in the plasma, whether it is stable in the human plasma or not, whether it is binding to human plasma components or not, and what is it basically doing. Because the peptide has been derived from the immune system, so it has to be present, work in the plasma only. So this is the point. Then we go for go no go decision. This is one of the critical parameter, or we call it as a stop point. Like any peptide which is to be taken forward or not is decided at this point. So normally we have 2 points like LRV and TV. TV is nothing but the dose at which the concentration at which the commercially available molecule is working. So for example, if we take antibiotic ciprofloxacin, it works at 2 microgram per ml. And if we take the nalidic acid, it works on 20 microgram per ml. So we have a point. So this is what this concentration. And if it comes to cytotoxicity, and ciprofloxacin has a cytotoxicity of 50. So this is the line that we put everywhere. And LRV is a concentration at which we decide whether to work on the molecule or not. So LRV, if any molecule's activity is before this, we stop, always we stop. But if any molecule which comes in this range, we always take that molecule forward for the clinical trials. So we have this go, no-go decision, and this is the critical step, number 1. And sometimes what happens is we try to make peptides, various forms of the peptides by using the replacements, and we always go back to the evolution, because changing every amino acid will be very much like if I change every amino acid in a 20 amino in a 4 amino acid stretch, then I have to make around 1 crore of individual peptides, and that is not practically feasible. So what we try to do is we try to look at what evolution has done. So normally in any given peptide sequence, not every amino acid is randomly changed. There are some amino acids which are conserved during evolution. So we always try to keep them as such because they are involved in the immune modulation or in immunomodulatory functions or antimicrobial function, whatever might be the peptide. So based upon that data, we always try to modify to get a better activity. Because as I told you, getting a higher therapeutic index is important. So we always made new variants or new molecules out of the older one, and we got a better efficiency or we got a better therapeutic index in our second case. And then we looked at how do these work on the bacteria? Do they kill the bacteria? Or do they create pores? So we looked at, as I told, we looked at the 2 bacterial strains, that is Staphylococcus aureus and Klebsiella pneumonia. Here we have done on all the 5, but I want to show you only 2 so that it's easy to understand. So if you look at the peptide, this peptide is bursting open the bacteria, but this peptide is making holes in the bacteria. So this is how they work. They normally, they either create holes so that the entire cytoplasmic content comes out, or they simply burst open the bacteria so that the entire thing is thrown out. Sometimes they normally create pores so that the entire cytoplasmic content comes out. So it depends upon the peptide to peptide whether they create pores or they just rupture the membrane. Whatever might be, in both the cases, they kill the bacteria. So once that is done, then we ask the fundamental question, is it a protein derived function or it is a random function? So what do you mean by protein derived? Means does my peptide is going to bind to a protein present on the surface, or we call it as a receptor? Is my peptide binding to a receptor, or it is simply binding to the membrane and killing the bacteria? So we use 2 different setups. One is the bacterial membrane made liposomes, and the eukaryotic. That means we have 2 lipids. One is the lipid that represents the bacteria, and then the other lipid which represents the eukaryotic lipids. So when we looked at it, our peptide is very specific. It only binds to the bacterial lipid, but it does not bind to the eukaryotic lipid or human lipids. So this shows that our peptide is not a receptor specific, and it's a membrane specific, and that membrane is also very specific. It only binds to the bacterial membrane, not to the human membranes. And then we try to check by other facts, that is flow cytometry also, to rule out any false positive results. So this is what the flow cytometric, where we have used the bacteria and we have added the peptide and showed that the peptide is binding to the bacteria, and it is binding to and it is creating pores. Then the second question is, okay, we have a peptide which kills the bacteria, but what happens if we try to put them on the human body? When we put them into the human body, what will happen? So normally when we try to put them into the human body, there are normally different type of cytokine response or chemokine response occur. So when you have a foreign body in your body, you have inflammation conditions, and we wanted to focus on how the inflammation is working on. So what we try to do is we have taken two different cells, that is THP1, which is nothing but macrophages, and then we have taken HACAT. HACAT cells are basically human epithelial cells, and then we try to treat them with E. coli LPS, and then we have added the peptide. We have not added the bacteria, because when you add bacteria directly to the cell culture, the bacteria will multiply very fast, so that you don't have enough time to study what is happening. And the second important thing is the plasma, the FBS will kill the bacteria, and they normally release the LPS. So we thought, okay, we'll take the E. coli LPS and Pseudomonas LPS, and then we added the LPS and peptide, and then we have estimated the what is the ELISA profiling and what is the cytokine gene expression, what type of chemokines are coming, what types of cytokines are coming, and what type of the things are happening in the cell when we treat our peptide, or what is happening when we treat with the LPS and our peptide. So basically it is like this. So if you have an infection, you have an LPS there, the bacteria is present there, and it releases the LPS so that it induces inflammation, and this inflammation leads to the downstream effects. So we want to see what happens when we add the peptide when inflammation is going on. So we added the LPS and we added the peptide. So why did not we take gram positive? Because when we looked at the immunomodulatory responses between the LPS and the LTA, which is the primary component of gram positive bacteria, the amount of immune response induced by the LPS is more than LTA. LTA is a weak immunogen. It does not induce inflammation when you have a gram positive infection. But when you have a gram negative infection, the inflammation is a bit higher because the LPS is able to induce the inflammation. And not only that, E. coli LPS induces a stronger response when compared to the Pseudomonas LPS or Klebsiella LPS. So for that, how did we do the cytokine profiling is by using a Procata multiplex immunoassay. This is one of the cutting edge technologies where you can screen all the 72 or 100 cytokines and chemokines simultaneously in a single cell. Like if I have, if you add LPS and then take the supernatant and then put them in Prakata multiplexing, you can know what all chemokines, what all cytokines that are being expressed. So they're basically, what is this Procata multiplex is basically Procata multiplex is as a bead, and on the bead they have an antibody which is of which is very specific. Like one antibody will bind to IL-6 and one antibody will bind to IL-2 and one will bind to MCP, and each of this bead is a different color. It is not, and they don't have the similar color. Like IL-6 binding bead has a different color than IL-2. And what we do is we simply, if you look at this one, the beads have the antibodies, and when we add the supernatant to the culture media, or when we add the culture media in which HACAT cells are the THP ones treated with the LPS as well as peptide is placed in this, the antigen will bind to the antibody, and then we purify them when we add the dye to them so that they bind to the secondary one. And once they have the binding to the antigen, and then we pass through them to two lasers. One is the green laser and one laser. We call this as Procata plex machine. So the green laser will measure whether the antibody is there or not. If the antibody is fluorescently labeled, that means there is an IL-6. If it is not there, then this will not be present. So you will not get any signal. whereas the bead color will tell what is it, whether it is IL-6 or IL-2. So this is basically a very cutting edge technology where within like 500 microliter of sample, you can identify around 70 different type of antigens that are present in your system. So based upon that, we have identified that when we give the LPS treatment and when we give the bacterial, the peptide treatment, there is a fluctuation difference in 4 cytokines, that is TNF-alpha, IL-10 and MCP-1. So the first thing that we looked at is whether there is any gene expression, whether there is any change in the fold of gene expression, because as long as there is a protein has to come, then it has to be a gene expression. But you have to remember, if there is a gene expression, never means that the protein will be present. If a protein is present, the gene expression has to be there. But if a gene expression is there, it never means that the protein will always be present there. So there is a bit yin and yang situation here. So first thing that we looked at is whether there is any protein gene expression is taking place or not. And then we looked at it. Then we did the ELISA to confirm like how much is the quantity of difference in there. And the results show that when we give us our peptide treatment, there is a huge decrease in the TNF alpha response. When we give the LPS, there is like the TNF is somewhere around 15,000 picogram per ml. And when we give our peptide, it falls down to 400 picogram. And we have looked at IL-10. IL-10 has no difference, which is good because IL-10 has a problem of propagating the inflammation. Then we looked at MCP-1. MCP-1 has to be very much because it's a inflammation marker, and IL-6 is also an inflammation marker, but IL-6 is a pro-inflammatory marker, so it has to get down, whereas MCP-1 is anti-inflammation, so it has to rise. So we looked at different markers, and then we asked like, okay, this is what is happening in THP1 cells. What is it happening with the Harcard cells? Because epithelial cells, because when I put my peptide into mice, the peptide goes through different cells. It goes through immune cells, it goes to epithelial cells, it goes through the mesenchyme cells, it goes through the muscle cells. And if it goes and induce inflammation in the muscle cells, then you have an issue. Your peptide should work only on the immune system. So once we looked at it, how it is interacting with the Harcard cells, and the data shows that the peptide does not interact with the Harcard cells. It is very specific and it interacts with only with macrophages. And all the other parameters are also same. It does not induce IL-6, it does not induce MCP-1, it does not induce TNF-alpha, it does not induce IL-10. So my peptide is very specific, it only interacts with the macrophages. So then we ask the question, okay, it is interacting with the macrophages, it's fine, but whether it is inducing the cytokine production or it is simply killing the macrophage so that the cytokine is coming out. So basically, we always look at cell death. Cell death, we have apoptosis, necrosis, and we have now pyroptosis. So we focused on pyroptosis because apoptosis and necrosis are very fast events. And whereas we have given our peptide treatment for 24 hours, MTT and LDH assay has shown that there is no killing of macrophages. So we wanted to see whether there is any pyroptosis happening or not. Pyroptosis is a cell death, but that occurs very slowly when compared to apoptosis and necrosis. But an important parameter in this one is when pyroptosis mediated cell death occurs, IL-1 beta is released. And we have looked in our Procarta Plus, we did not find any IL-1 beta expression. We have only found TNF-alpha, MCP-1, IL-10 and IL-6. So we went back to our and then we checked it whether there is any IL-1 beta production, or whether if what happens whether there is any pyroptosis occurring or not. So when we looked at this one, we have observed that our peptide don't kill the cells and it does not induce any pyroptosis. So that was very good. Now we know that my peptide works against bacteria very well, and it does not do any significant damage to the human body. It does not, it interacts with the immune system in a positive way, not in a negative way. So once we are done with that, then we started to look at the applications of it. Then we went for the in vivo studies, where we have looked at is how fast the bacteria, how fast the peptide works. The my peptide kills the bacteria within 2000 seconds. That means roughly around in 10 minutes, it kills the bacteria. So this is how this is the negative control, and this is the peptide, like how the peptide kills my bacteria. Like we measure the cytoplasmic contents that are coming out and how fast it comes out. And we realized that within 10 minutes, my almost all, like the bacteria kills the entire bacteria, 90% of bacteria. And then we looked at the how much is the bacteria that is present on the surface when I give the treatment. So normally there is 100-fold bacterial log difference. So this is very good. And then we looked at the whether we can very specifically kill the bacteria present in human body, or if the bacteria is present somewhere, we can kill it. So what we did was we looked at the infection on the lenses. We have taken human contact lenses and inoculated with the bacteria, and then we added the peptide, and after 24 hours we cultured those lenses to see whether any bacterial is surviving or not. And our results show that our peptide can kill bacteria wherever it is present. It can be on the contact surface, it can be on the catheters that we use for human infections, and it can work on the surface of the skin surface. Wherever it is there, it works. So then we looked at it, how does it work inside the human body? So we injected the peptide into human lungs and checked what is happening. When we added the LPS, there is a lot of infiltration, but when we added our peptide and LPS, there was no infiltration of cells. These are the neutrophil cells, WBC, RBC, and everything is coming up. There's a huge damage of the linings and everything, but when we add the peptide, there is nothing is happening. And then we looked at the total profiling, like whether there is any difference between the human macrophage and the human mice body, because in the mice immune system and human immune system are bit similar, but there are fine differences also, to see whether there is anything. Like when we looked at it, we have identified that in humans we did not find INF gamma production, INF alpha production, but when we injected our peptide into the mice, we have found that there is an INF alpha induction. But when we give the treatment with the peptide, it is getting down. And then we looked at the production of TNF alpha and MCP1. We have identified that the MCP1 is still produced, TNF alpha is getting reduced, and there is no hemolysis of the mice blood cells. So it is safe in the peptide is safe in the mice, and it does not kill the mice. So whatever the phenomenon that we are observing is because of the killing of the bacteria, but not because of the mice. So in nutshell, what we did was we identified a peptide sequence and demonstrated the activity against the different pathogens, bacterial pathogens, and we have demonstrated that the peptide works in the mice. It kills the bacteria present in the mice. And we have shown that no physiological damage occurs. There is no difference in the cytokine profiling, or there is no difference in the immune system. So this is what we have done till date, and now we are looking for the various data, like what happens in the human component. But I cannot share the data at this moment because it is unpublished. So this is what we do on routine basis every time. So till date, we have identified cryptic peptides, or the peptides from all these proteins. And what are we planning to do it in the future is to see the synergistic effects, like synergistic antimicrobials for reducing the side effects. So when we, like I told you, when we have used the peptide in the human cell system, we did not get INF gamma or INF alpha, but when we injected into the mice, we had the INF gamma production. So we want to see like whether if we use a combination of two different peptides, whether we could reduce the side effects. And we are looking for better bioinformatics tools where we can still reduce the peptide and have the efficacy. And one of the area that we are trying to look is to have a novel peptide delivery system for sustained release. And so this is one thing that we are working on right now. And what we want the next generation of people to work on is how the host defense peptides interact with different cells, like how it interacts with neutrophils, monocytes, mast cells, and dendritic cells, and T and B cells. This is one thing that is not explored in host defense peptide, even though we are directly doing our preliminary studies with monocytes or macrophages or with neutrophils. And then we are simply going into the animals, but we have never studied in detail like what is happening, how my peptide is interacting with mast cells, there is any difference or not. If I put it in other words, I have both MCP1, IL-6 and IL-10 production, but I don't know whether that IL-6 is coming from monocyte or from the mast cells or from the neutrophils. From where it is coming, I don't know. So that needs to be explored in detail. And that is one thing that that other people can take care of. And one of the other major issue is the animal models, like what happens when I inject my peptide into human brain or some other brain location. I don't know how the peptide behaves in the intestine also, I don't know. And what should be the host factors, like what happens when the peptide is injected into very old people or very newborn infants or with the people in the pregnancy stages. And these all things are to be explored. And these are the next generation challenges in this area. And one of the major area is how a peptide behaves in the mice, rat, and monkey and dog, because these are the four animal models that FDA asks you. You have to show your peptide activity in any of the two animals. Like if you show in mice, you have to show either in monkeys or in dogs. If you have shown in the rat, also you have to show in monkeys or rat. So you have to show it in two different higher animals or in two different models. And then one of the major issue is the cycle, the intensity. Like in humans, neutrophils do not come so fast, but whereas in mice, the neutrophils come very fast. Whereas macrophages slowly pick up and then they stay for like weeks and neutrophils go down. And we really don't know how is the cycle in the dogs and in the mice when we give the peptide, whether this particular model is based upon the human system, but we really don't know whether this is the same in this one or not when we give a peptide treatment. So these are the two areas where people can start working on, because host defense peptides have been already saturated. There are a lot of people working in the line, but most of the people who are not working in this line are these two areas where people are not working very much. And other area that is recently coming up, if you are not interested in host defense peptide, but if you are interested in immunology, then one of the area where you can work is the nanoparticles. We really don't know how the nanoparticles will react with the immune system. Like what is the immune functions of the nanoparticles? Like different type of nanoparticles are coming up, like rods, loaded ones, and the tagged ones, and there are different type of nanoparticles with the iron in composition or silicone, like how the human body will react, because the preliminary data is quite confusing. Like for example, if you have taken silver nanoparticles of different sizes, the higher is the size, the lower is the loss. But higher is the size, higher is the cell killing. Like if you have the higher cell size, the cells will die very faster. So if you have a smaller size, they are excreted very fast, but the cells are safe. So you have to find a narrow point where the size and the staying in the body has to be explored. Like if you have a bigger nanoparticle, they are eliminated very fast. If they are smaller in nanoparticles, they are killing the cells. So you have to find a balance between them and how these nanoparticles will interact with immune system, whether they produce cytokines, chemokines, pro-inflammatory, anti-inflammatory, or how is the cycle. This is one area that is upcoming, and most of you can start thinking about this in the line. And one of the other area, if you are interested in exploring the basic science, then one of the other area is to explore how the pathogens bypass the immune system. Like normally pathogens, like for example, some viruses make membrane membrane complex or MHC inhibitors, so that the MHC complex does not form. So the virus infected cell will go on multiplying. And there are some which makes the cytokine chemokine binding proteins which bind to cytokines and chemokine. So if you are interested, you can look at different parasites, fungi, and bacteria. How do they bypass, or how do they fool the immune system? This is also one of the new area that is coming up, like host pathogen interaction. We call them as host pathogen interaction. And this is what I want to tell you. And before I close, I would like to thank my students who are working on different areas that are related to my group, and my collaborators who help me, and my funding agencies which run my lab, like the CSIR, which is the primary source of my funding, and then MOES, DST, and DBT, and my collaborators for helping me in the issues. And the conclusion is very simple, like multi-drug resistance is coming up, and it is a serious concern. And proteins and peptide therapeutics not only cure infection, but they work with the host immune system. So you have lesser side effects. And then you have the protein and peptide therapeutics speed up the recovery process and minimize the host damage. And host defense peptides are having multiple functions, so they can become a therapeutic molecules, and host defense peptides will be the next generation biotherapeutics. And this is thank you. And now I am ready to take your questions.
There was one question from one of the students. What are QSAR models?
QSAR is quantity. QSR, right?
QSAR, she said.
So the QSR is nothing but quantitative structure activity relation, like what happens when I replace an arginine with histidine, or when I replace a valine with other amino acid, what will happen? So this type of studies are called as QSAR studies.
So Manoj sir, it's now your.
Okay.
Manoj sir.
Okay, madam. Okay. Thank you, Mukesh, for your nice and wonderful lecture. And your topic was apt for the title of our seminar, Cutting Edges in Molecular Immunology. Cutting edge technologies in the molecular immunology. And you have thrown light on host defense peptides, immune modulators, quorum quenching, small molecules as antimicrobials, and antimicrobial resistance mechanism offered by pathogens. Now, the bacteria, whatever bacteria you were taking, 4 different types of bacteria were taken for your study, and you have explained how disease burden profiles occur, and how antimicrobial resistance screening is done, and how can a bacteria protect itself from the antibiotic it is producing. Now, this was very nice to hear from you about Casper technologies, bacteriophages, and nanoparticles, which are the coming technologies to kill microorganisms. Peptides and proteins, we have talked about immune protection against normal flora, where the topic of interest, where our MSc, both microbiology, biochemistry, and biotechnology students are within their course structure. They study about immunity and biopharmaceutical products, and which serves to be highly efficient particles, post-defense peptides, and you have definitely talked about your work plan and therapeutic index, and definitely your future studies need to be addressed by the research scholars of the department or the students, and we hope you also will be more successful in creating such small peptides in gaining resistance against disease-causing agents. Thank you, Mukesh. Thank you, ma'am.
Thank you, Mukesh.
Thank you, ma'am.
Professor Sreelatha madam, you can propose the vote of thanks because this is the end of our first session. Just unmute yourself, madam.
Good afternoon, all. I feel privileged to present vote of thanks on the occasion of international seminar organized by Microbiology Department of Chaitanya deemed to be University. On behalf of our university, our microbiology department, and my own behalf, I extend my hearty gratitude to all the speakers for gracing the occasion and sharing their knowledge with us. Today, we had an opportunity to hear your informative and interesting lectures, and this will surely be going to help our students in understanding the complex topics. Really, you made it so easy to understand. In spite of your busy schedule, you have taken out time to enlighten our students. We are very thankful to Divya, Priya madam and Mukesh sir. I would like to thank all the guests and participants for their active participation. I want to extend my generous thanks to all my colleagues, scholars, and students for their support. Finally, I would like to thank our Chancellor sir for his constant support. Without his support, it's not possible to conduct such an event. Last but not the least, thanks to all the people who helped us either directly or indirectly to make this event successful. Thank you. Thank you, one and all. Madam?
Thank you, madam. We shall now. Oh, yes, ma'am.
Ma'am, at 1:30 we'll again meet for.
Madam, we are going to meet at 2:30, madam.
Okay, madam. Okay.
So I request all the participants and those who are presenting your PowerPoint presentations and all the others who are listening to this webinar to again join at 2:30 with the same link. You can join with the same link. So at 2:30, we'll again start the second session. And for the second session, Professor Christopher sir is the chairperson and Professor Suma is co-chairperson. So each student will be given, or each scholar will be given 15 minutes to present their presentation. So you can share your PowerPoint presentation, after which we'll have a small discussion and we will continue this session at 2:30. Thank you very much.
Thank you.